Hey guys, welcome for chapter 12. This is going to be on the cell cycle specifically. We'll look at the cell cycle and mitosis. So here we go. The continuity of life is based on reproduction of cells, whether that be the reproduction of an entire organism if you're a single-celled organism, or replacing damaged cells or creating more cells for growth purposes if you are a multicellular organism. But all organisms need to be able to reproduce. Okay, reproduce cells, reproduce in general. It's one of the characteristics of life. So again, for this chapter, what we're going to be really looking at is mitosis, which really allows both growth as well as reproduction if you're a single-celled organism. And it can also allow for growth and development, and repair is another good example for mitosis. Key is what we're looking for. Here we go. There's those. The key is that they're looking to duplicate, and a key word here is identical. One of the things that we'll do in this unit of cell division, we're going to learn about both mitosis and meiosis. However, we really look in depth at the differences between the two. And a key difference is that in mitosis, which is what we're talking about in chapter 12, we create identical cells. If you think about why that might be the case, well, if you think about the general process, why we're doing this, growth, repair, if you get a cut in your skin, I don't want liver cells to fill in that gap. I want my exact same skin cells to fill in that gap, right? So that is just one specific example of why they need to be identical. They need to be able to perform the exact same function of the cell that was there before this. So that's a specific example with repair. But if we're talking about growth, going from a child all the way up to an adult, you're going to want to create more cells, right? And those cells still have to do those exact same functions. So that identical piece is very important. You absolutely need to understand that in mitosis, which is for growth and repair, development, we create identical daughter cells. Absolutely essential knowledge right there. Because when we look at meiosis, that will not be the case. We'll talk about how meiosis creates variation. Very different. Okay. They both are cell division, however, they have very different purposes, functions. Okay, so identical cells is very important. Cells will duplicate their genetic material in order for this to happen. So how are they becoming identical? Well, it's because they have the exact same DNA. Okay, so very important that they can do this. Why does the DNA need to be copied? Well, we're making two identical daughter cells. Each of those cells need to have a full set of DNA, so it needs to be replicated before any division can happen. So when we talk about the cell's DNA, we call the full set its genome. In an average human cell, that's about 2 meters worth of DNA. So quite a bit. And in order to keep them organized and make it easier to replicate them, what we do is we package that into chromosomes. So it's just chromosomes are basically condensed DNA in order to help keep it organized. In humans, we have 46 chromosomes. Each species really has a characteristic number. Ours is 46. You get 2 sets of 23 to make up that 46. In other words, you got 23 chromosomes from each parent. And when you bring those together, you have 2 sets of each, basically a pair of each chromosome. There's 23 different chromosomes, but you have a double set. We call that diploid. And all of your somatic cells, in other words, your body cells. So every cell in your body, except for your gametes. So if you're female, that'll be your eggs, and if you're male, that will be your sperm. Every cell except for those gametes has that full set of 46. The gametes would only have 23, but we'll talk more about that when we get into meiosis. So eukaryotic chromosomes are going to consist of chromatin, and chromatin is a more diffuse. Here's how I kind of think of this. When your cells are in interphase, which is the first phase of the cell cycle, we'll get into that in a little bit. The DNA in your cells would be in this, it's called chromatin. And how I kind of think of that is it's like a bowl of spaghetti noodles. So if you think about, picture in your brain right now, a bowl of spaghetti noodles. It's very diffuse. It's not real well organized. So what has to happen for cell division is we have to take that chromatin, that spaghetti noodle, and we have to condense it into chromosomes. Okay, and that's, again, the main reason for this is for organizational purposes. So let me get to a picture. If we look at this, if you look at this in comparison to, again in your head, picture a bowl of spaghetti noodles. This is extremely organized in comparison. But it's essentially made up of exactly the same thing. What essentially this chromosome is it's a bunch of spaghetti noodle rolled up really tightly around proteins, making it organized. That's what we have to do in preparation for cell division. DNA is replicated, and then the chromosomes will condense. Why do they need to condense? It's just for organizational purposes, honestly. So DNA would have to get replicated. Why does DNA have to get replicated? Because remember, we're making two cells at the end. Both cells need a full set of DNA. So that's two parts that have to happen. So this would be, just keep in mind, this is not what that chromosome would look like before it's replicated. It's just a model showing you what's happening, kind of. Remember, this would actually be chromatin, that spaghetti noodle. But if we had that spaghetti noodle really organized pre-mitosis, that's kind of what they're showing you. But they're showing you have one set. That one set gets duplicated. Now you see a double set here. And then at the end of mitosis, you separate that to each cell. But again, look at this cell in comparison to the original cell, and it's exactly the same. So that's just kind of an intro to kind of how this is going to work. But eukaryotic cell division consists of both mitosis and cytokinesis. Cytokinesis is the division of the cytoplasm. Mitosis is the division of the nucleus. So how I kind of think of that is mitosis is kind of how we separate the DNA, and then cytokinesis is how we separate the actual whole cell itself. We will talk in the next chapter about meiosis. That'll be chapter 13, so that'll be a separate presentation. But meiosis is very different. There is some commonalities between the two that we'll talk about after we've looked at both. But meiosis' main job is for sex cells. So in a multicellular organism, that's going to allow for reproduction. But mitosis, on the other hand, is all about creating body cells. So pretty much every cell in your body, except for your sex cells, is using mitosis. All right, so let's look at that mitotic phase then. But first, we need to look at the cell cycle. So this is really important. It's actually the title of the chapter. The cell cycle is going to consist of the mitotic phase, which is, if you look at this pie chart, this phase right here. Okay, but you notice most of the cell's life is spent in this other phase. It's called interphase. Okay, in interphase, we can break down into three phases. We have the G1. G stands for growth for the most part. Then we have the S phase. S stands for synthesis. So what's going to happen there? DNA synthesis. This is where those chromosomes would get duplicated. Also, all the organelles in that cell would get duplicated. You think about why that must be the case. Again, we're creating two cells at the end of this. Both cells, they don't only need a full set of DNA, they also need a full set of organelles so that they can perform all the functions. So that would all happen in the S phase. And then that G2 phase finishes up interphase. So we have G1, S, and then G2. Again, G we think of for growth, S is for synthesis of both DNA and the organelles. So there's really two parts to the cell cycle. You have interphase, which the cell is growing, synthesizing its DNA and organelles. And then you have the mitotic phase, which is where we actually divide. Keep in mind, very important, the interphase is what a cell spends most of its life in. So interphase, again, can be divided into those three phases. We have G1, G stands for growth, S, synthesis of DNA and organelles, and then the G2 phase. The mitotic phase is both mitosis and cytokinesis. So again, mitosis, division of the nucleus or the DNA, and then cytokinesis, division of the rest of the cell. And that we would really break down into 5 distinct phases. Some people would say 4 distinct phases. When you are a freshman, this is probably not a phase we talked about. The pro-metaphase, it's kind of a combination between prophase and metaphase, obviously hence the name. But we'll go into exactly what happens. Just keep in mind for your test, you can never lose points for not knowing what pro-metaphase is. Okay, so just keep that in mind. So if you don't end up really diving in deep and learning about that pro-metaphase, it's not the end of the world. It all starts with prophase. And I think of P for prepare. This is preparing the cell to divide. So what has to happen here? Several really pieces that will happen in prophase. Specifically the DNA, the chromatin. Remember that you can see why I called it spaghetti noodle, right? Here's interphase. This is a picture of a cell in interphase. And what you're seeing here is this purple, that's inside the nucleus, but that's all DNA. That's your chromatin. You can see, again, why I called it spaghetti noodle. Okay, what has to happen in order to enter prophase is look at the difference in the DNA. We went from a spaghetti noodle, now we have distinct chromosomes. So I think of prepare, P for prepare, right? Because the DNA is turning into chromosomes. In other words, that DNA was condensed into chromosomes. Other pieces that happen in prophase, you'll see these centrioles, centromeres. Sorry, not centromeres, centrosomes, which is an organelle that aids in cell division. They're moving to opposite poles. You can see it in comparison to right here. They have those centrosomes are moving to opposite poles. Other thing that happened, you can see it's basically happened by this, but this nuclear membrane will dissolve. So we'll see that as well. You can see that it's no longer there here. So that nuclear membrane dissolves, the DNA turns into chromosomes, and the centrosomes are really moving toward the poles. Key thing that happens in pro-metaphase, and sometimes, again, sometimes we just lop these two together, okay? So if you learn pro-metaphase as part of prophase, it's not the end of the world. But what you're seeing happen here, at the centromere is that part, if I go back to a picture of a chromosome, these parts where the two chromatids come together, you can see right here, it's called a centromere. So just be aware that's a term you should probably be aware of. We have two sister chromatids here. They are identical. Each side of this is identical. They're held together at what we would call a centromere. Now, on that centromere, on each side, you can't see it in this picture, but there would be what is called a kinetochore. So you can see here is that term kinetochore. And they are, you can see it's pointing toward the centromere. They're on each side of the centromere. Now what happens is here we have a spindle fiber. Spindle fibers arise from the centrosome, and they're kind of would do a lot of the work as far as lining up these chromosomes and pulling them apart eventually. But that spindle fiber is going to attach to a kinetochore. And there'll be a better picture of this later in the presentation. But again, at prometaphase, what is happening is the spindle fiber is attaching to a kinetochore. Essentially, it's just attaching itself to the chromosome, each side. Okay? Now, that's going to lead us to metaphase. Now, you might wonder, well, why do they line up along the middle? And it's because it's there's a microscopic tug of war happening here. You think about, we have a spindle fiber attached to each side of this chromosome, and those spindle fibers are equal in their strength. So when what happens in a tug of war when you have both sides at equal strengths? Well, essentially you have a stalemate, right? And what happens is the flag that would be in the middle of that tug of war stays right in the middle. That's why the chromosomes line up along that metaphase plate, the equator of the cell. It's because these spindle fibers are actually pulling equally in both directions. Now, one other thing that I just want to point out to you is you'll notice that not all of the spindle fibers have attached to a chromosome. For instance, look at this one and here. Okay, so they actually attach to each other. So two spindle fibers attach to each other, and they actually push against each other, and they elongate the cell. So if we look at this cell in comparison to this cell, you can see that the cell is starting to elongate, and that the reason for that is these spindle fibers that have not attached to a chromosome are pushing against each other. So if I just go back, prophase, the DNA condenses into chromosomes, the nuclear membrane will dissolve, and the centrosomes move to opposite poles. Prometaphase is when the spindle fiber actually attached to the chromosome at the kinetochore. Metaphase, they line up along that metaphase plate because of a microscopic stalemate of tug of war. Now we get to anaphase. Well, before I do that, I just want to point out M. The M in metaphase, you should think middle. Think M for middle, or midline, because they line up along the midline. A, anaphase, think A for apart or away. Okay, and why would we call that? Well, look at the chromosomes. They've now detached at that centromere, and they're being pulled to opposite poles of the cell. They're moving apart from each other. Okay, so each side, remember, these two parts are exactly identical. They are copies of each other. So each, eventually when we start to see two cells here, each of those cells is getting an identical copy because we've pulled them to opposite parts in anaphase, A for apart. The last phase is called telophase, telophase sometimes called. Now how we think of T, I think of T for two. Sometimes I think of T for twin. Why would I call it twins? Because again, we're creating twin cells. These are identical cells. What happens in telophase? We start to see two cells. You can see the nuclear membrane is reappearing. We're going from distinct chromosomes. You can see the DNA is diffusing back into chromatin. So lots of stuff happening in telophase, but essentially we've created two identical cells. Now, we get to cytokinesis. I'll show you this in a minute, but cytokinesis, it looks like this. So cytokinesis is the division of the cytoplasm. Now we have two identical cells. Go back to those videos. I want to show you both of these quickly.
In mitosis, the nucleus divides to produce two nuclei that are genetically identical to each other.
Now, while he's talking here, take note of this picture here. What you're going to see is this DNA is going to get replicated.
And the parent nucleus. To prepare for division, the DNA replicates in the preceding interphase. Although the chromosomes are not yet compacted and visible as discrete bodies, we illustrate them as such to show the formation of a second chromatid per chromosome.
So again, now you can see that there's two on both sides of this thing, again, are identical copies.
Also, during interphase, the cell replicates its centrosome. Centrosomes are microtubule organizing centers, which are important for chromosome movement during mitosis. In many organisms, each centrosome contains a pair of centrioles. In the first phase of mitosis.
Now, here we go. We're going to move into prophase now. But take a look at this DNA. This red here is the DNA. You can see why I called it the spaghetti noodle.
On prophase, chromatin coils and condenses, resulting in compacted chromosomes that are visible with a light microscope. In prometaphase, the nuclear envelope breaks down and microtubules emanating from the centrosomes attach to the chromosomes.
Okay, so now we have no more nuclear membrane. Remember, sometimes we would include prophase and prometaphase together, but one of the things that you can see here is the spindle fibers have now attached to the chromosomes at the kinetochore.
Microtubules attach to structures called kinetochores, which are found in the centromeres of the chromosomes. In metaphase, the centromere regions connecting paired chromatids become aligned in a plane at the cell's equator.
M for middle, you can see why.
In anaphase, the centromere pairs from each chromosome separate, and
A for apart, again you can see why.
the single chromatids, which are now considered individual chromosomes, move toward the poles. In telophase, the separating chromosomes reach the poles. The division of the cytoplasm, called cytokinesis, also begins during this phase. As the cells enter the next interphase, the nuclear envelopes and nucleoli reform, and the chromatin again becomes diffuse.
So again, you can see why we call it two cells, twin cells. You can see the DNA reforming into that chromatin, that spaghetti noodle, and then the nucleus will reappear. All right, one more quick video to illustrate it.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes.
So we're starting prophase. By the way, this is a nucleus here. We see that DNA is now in chromosomes, so we are entering prophase here.
Meanwhile, protein fibers grow from the migrating centrioles.
Centrosomes are moving to opposite poles,
forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole.
Now what you're seeing here, this is a spindle fiber. So they're showing a really good image here of how that spindle fiber, and this is a kinetochore right here. So we have the centromere where the two come in contact, and on each side of that we have a kinetochore.
Itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles.
This is anaphase.
Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes that
Now here will be telophase.
unwind and the nuclear membranes reform.
So there you go. Good examples of videos. If feel free to go back and have access to these slides. So, all right, just to review a couple things. The mitotic spindle, that would be the spindle fibers along with the centrosomes. And again, they're really a driving force in this mitosis process because they're going to really move those chromosomes, move the DNA in essence, to where it needs to go to make twin cells, two identical cells. They do arise, the spindle fibers arise from the centrosomes. So we see that here, okay? And some of them will bind to a kinetochore on a chromosome, but others push against each other to elongate that cell. You can see again some spindle fiber attached to a kinetochore here. Some of them are elongating the cell by pushing against each other. All right, so cytokinesis then. On the left here, we have animal cells. On the right here is a plant cell. What we call this is a cleavage furrow in an animal cell. And what that essentially is, it's a contractile ring. It's actually made by actin, which is a part of your muscles, which allows them to contract as well. But essentially, we have a ring that pinches this cell into two. That's for animal cells. In plant cells, because they have a more rigid cell wall, you can't pinch them into two. What actually happens is they grow a cell plate between them. So we went from one cell to two cells here because of the plate that was grown between them. Let's see that again here. This is just an actual look at, it's obviously not quite as clean as what those made up images look like. This would be an example of prophase and into prometaphase. So you can see, look at the difference between the DNA, the more diffuse into now distinct chromosomes. Here we have your metaphase. They're lined up along the middle. Anaphase, they move to the opposite parts. And now you're seeing, this is for a plant, that cell plate is actually going to split these into two cells. Now, there is another form of cell division that bacteria do. This would be prokaryotes. Remember, they do not have a nucleus. We actually call it binary fission. It's somewhat similar. They don't have the distinct phases like mitosis does. But in prokaryotes with bacteria, what they'll do is they have a circular DNA. So you can see that here. And they still have to replicate that DNA, and then they split that way. So just you should know the term binary fission. We don't nearly go into as depth of coverage of that, but this is essentially cell division in prokaryotes. I'm going to skip ahead a little bit. All right, so what do you need to know? You need to be able to describe the events that occur in the cell cycle. So that would be interphase, all the three phases of that, G1, S, and G2. And also the mitotic phase, which is mitosis and cytokinesis. And you need to be able to explain how mitosis results in the transmission of chromosomes from one generation to the next. Okay, and that was through those phases, right? So you should be able to explain the phases of mitosis: prophase into prometa, into metaphase, into anaphase, into telophase. And be able to, because if you can explain those, you're essentially explaining how those chromosomes get from that original cell to the next. All right. We also need to go through how the cell cycle is regulated. And essentially, it comes down to a series of checkpoints. So the frequency of cell division varies on the type of cells. There's some types of cells that will never divide after you've reached maturity. Muscle cells are an example of that. Neurons are another example of that. But for other cells, like skin cells, for instance, they're always going to need to divide. Okay, so differences in that way. But here we go. One thing that there is cytoplasmic signal. Essentially, this is chemicals that are involved in the process. So what this is showing here, if we fuse two cells together, so what they did in both of these experiments is they fused two of these cells together. And when we do that, both cells, you see, end up becoming in the same phase. Now, what's the reason for that? The reason is because there's chemical signals in the cytoplasm that really trigger a cell to enter certain phases. So, for instance, when this G1 cell got that chemical because it fused with this cell, it jumped immediately into the mitotic phase. Same thing over here. This G1 cell jumped into S phase because now it had those chemicals. Okay, so just be aware that there's chemicals that are involved in this process. There's also checkpoints. So you can see there's a couple checkpoints. We think of this kind of as a clock. So after you've passed the checkpoint, you move into the next phase. For instance, here's a G1 checkpoint. If the cell passes that, it's going to enter into S. And then eventually it'll pass that mitotic phase, and then eventually it'll hit again another checkpoint. It's basically kind of like just checking, are you ready to go to the next phase or not? Sometimes, this is a really, really important checkpoint. Really, the only one that I specifically talk about in class, actually, is that G1 checkpoint. So this would be more at the end of G1, that growth phase. Are you ready to move on, or are you not? Or are you a cell that doesn't need to divide anymore? Like that neuron, that muscle cell that I mentioned earlier. Let's say that cell is a muscle cell or a neuron, and once you have enough of them, you don't need any more once you've reached maturity. then that cell can sometimes go into what's called G0, and that is a non-dividing state. So all those neurons that are in your body, the muscle cells that are in your body that are no longer dividing, are likely in that G0 state because they don't need to divide anymore. Remember, cells don't and organisms don't want to waste resources. So if we don't have to make more of them, we won't. We'll go into that G0 non-dividing state. And what really will determine that is that G1 checkpoint. But once you've passed that G1 checkpoint, you're almost assuredly going to divide. Now let's think about why. What comes after G1? Well, it's that S phase, right? So why are you really likely to divide after you've passed this checkpoint? Well, in the S phase, we're duplicating all the organelles, we're duplicating the DNA, and that's not necessary unless you're going to divide. So once we've passed this G1 checkpoint, we're really, really likely to divide. Now, what really determines do you pass the checkpoint or do you not? There's two chemicals that are involved, and I'm not going to ask you to know specifics on cyclins or CDKs at all, but you should know that these are the two chemicals that basically regulate the cell cycle. Okay, that's it. That's all you need to know about them. Is they are chemicals that again regulate the cell cycle. So if we look at this graph shows where they're high, they fluctuate throughout the cycle. But again, you don't need to know when they're high, what they do even. All you need to know about cyclin and CDK is that they are chemicals that help regulate the cell cycle. Are we ready to pass that checkpoint? Are we not? Cyclins and CDKs determine that. There's other things that would be involved with it. Growth factors, for instance, is one. I find this pretty interesting, especially when we compare it to with cancer. One thing we have is in growth factors is look at this culture without growth factor compared to with the growth factor. You'll notice that the cells will fill the bottom when the growth factor is present, but when they don't have it, they don't divide at all. So growth factors are important in order to allow those cells to divide. That would be an example of an internal signal. We also have density-dependent inhibition. This would be external signals. What this is kind of showing you here, we have a culture, and this you can see cells have completely covered the bottom. And what they did here is they scraped out a small area. And you'll notice in this last picture, that scraped out area is filled in, and the reason is because cells have density-dependent inhibition. Once they're fully crowded, they'll stop dividing. And they also need to be able to attach to something, so they had something to attach to in that area. Normal mammalian cells, once that area is filled, they will stop dividing. However, we compare that with cancer cells. What you're seeing here is they exhibit no density-dependent inhibition. So again, once that scrape was filled, they just kept dividing. They also do not exhibit anchorage dependent. In other words, they don't need anything to attach to. They just keep dividing and keep dividing. How we define cancer is cells dividing out of control, essentially. They've escaped the controls of the cell cycle. So even though they have a full layer, even though they have nothing to attach to anymore, they continue to divide. So cancer cells do not respond normally to the cell's control mechanisms, and because of that, they'll form tumors. They're also pretty much immortal. This is a crazy thing about cancers. Normal body cells will only divide about 20 to 50 times, and eventually they'll stop and die. However, cancer cells, we've in science basically found them to be immortal. In fact, we took cancer cells they're called HeLa cells. The reason why they're called that is we took them from a woman, Henrietta Lacks, in 1951. They took cancer cells from this lady. They are still, to this day, almost 70 years later, still dividing in culture. They still work. Okay? So that's just crazy. They're almost immortal, and we find that they don't die. So when cancer cells continue to divide, they make a tumor. So if we look here, what we essentially have is a tumor of cells dividing out of control. Now, why does that cause problems, you might wonder? Well, there's a couple different types of tumors. We can have a benign tumor. Benign tumors are those that don't really cause serious problems. They can usually be removed. They haven't invaded anything. However, once they start to become what we call malignant tumors have now invaded tissues. And if a tumor has invaded tissues, that tissue can no longer do its job. And that's when you start to see cancer causing problems. And when cancer starts to become a really, really big issue, you've probably heard about like stage 4 cancer. Stage 4 cancer is when cancer has started to metastasize. And what that means is it's now traveling, it's going to other spots in the body. So maybe that's through the lymph, maybe that's through the blood, but the cancer cells are now moving throughout the body. Cancer is always named for where it originated. So if you hear somebody has lung cancer, that just means that it started in the lungs. But you can have lung cancer in your brain. Okay, so what has happened in that case is that lung cancer has metastasized and transported to other parts of the body. And that's really stage 4 is where cancer starts to get really hard to treat, and obviously a sensitive subject for pretty much everybody, but important to be able to understand how that relates to the cell cycle as far as this class goes. So hope that's helpful.